So, as far as I know, this is the first ever longevity science news podcast where somebody just walks through new scientific findings and papers of the aging field. How will this go? We will do a deep dive through 5 recently published papers that are relevant to the longevity industry. And this is not going to be another eat this, do that podcast. This is about the science and about the industry, and it is made for people actually working in the industry. So if you are a scientist, engineer, if you do anything, if you want to contribute towards this fight against aging, this podcast is for you. And if you just generally really want to learn about the science and not just the day-to-day, or you should take this amount of creatine or whatever, or this amount of NMN, or you shouldn't take NMN, this is not what this podcast is about. Before we begin, I wanted to make a note that I want to make this relaxed. Just lean back, enjoy. Imagine you hang out with a friend that is telling you about what he learned about longevity. If you don't want to relax though, I also made timestamps down on YouTube, or I think also on Spotify, and you can just jump to the topic or to the paper that you are interested in. And but if you want to go relaxed, as I said, just lean back and stay with me as I go through this. I wanted to make some tea, but it's super hot, so I just got myself some water. So let's begin. Or before we begin, who am I? My name is Elias Schlie, and my goal is to make aging and death completely optional. For that, I'm currently working really, really hard on building a skill set that contains, of course, really strong understanding of biology, to contribute towards longevity, really basic, really solid understanding of biology, really solid understanding of AI and machine learning, and ability to write programs that could help. And I decided to build this because this podcast, this longevity news, is something that I would love to have. Like in my research, I looked around and I found a lot of newsletters, and I thought oh, that's so cool. There are people that just take the time, they go through what is new in the field, and they make newsletters, and then they publish it, and then I can read it. And I signed up for all those newsletters, but turns out that I don't read all of them. But what I do is I love to listen to podcasts, and I was like, I would love if there would be a podcast where somebody just walks through new findings in the longevity industry. It's like a newsletter, but in a podcast format and in a chill format. There's just a mix of an audiobook, a person talking to a friend and a newsletter. But because I couldn't find that, I now trying out if I want to do it myself. So this is just still an experiment. This is the first time I'm doing this, and depends how much I like it. I will continue doing that. For now, what I can say is that it's a lot of effort. You might think, it's just 5 papers, that doesn't take so long, but yes, it takes very long. If you I really tried to deeply understand those papers to make a good presentation, and all this, I think also specifically because it's the first time, it just takes a long time. And also it's not just those 5 papers that I looked at. I looked at way more papers. I decided for those 5. Although I wanted to say that because it is the first time, it's a bit of a random and lucky draw of 5 papers. And it's just because in the beginning, it's very hard to see, what is exactly the news that is really the new creme de la creme of the papers. But if I will continue, when I continue to do this, I promise that I will give my best to get better and better at choosing the papers. And even the 5 that I chose this time, I think they are really interesting papers, and I really like them. So there's definitely some value in this first episode. Let me take some water and then we can continue. Okay. Let's go through the topics, through the 5 papers that are on the menu today. The first one is about the Rodent Aging Intervention Database, short RAID, which is a new database. The paper is by Pitu et al. And the infamous man with a long gray beard, Albie the Gray. Yeah, this is the first paper. Second paper is about a high-throughput yeast aging study. It's called Yeast Aging from a Dynamic Systems Perspective, and it was written by Mobaraki et al. I think this is how you pronounce it. Third paper is about metabolic digital twins. It's called Optimizing Metabolic Health with Digital Twins by Su et al. And yeah, about how we can use Metabolomi, kind of map it to build digital twins. I will talk more about it when it's when we're at this paper. Paper number 4, how cities can be used to learn about aging and exposome influences, or how aging is influenced by the exposome. Exposome is essentially everything that comes from the outside onto your body. So it's like the air you breathe, the food you eat, the like just what you're exposed to. By Woods et al. And yeah, this paper, just a bunch of known names in the longevity industry. Like, I'm not sure, like, I know Andrea Meyer was in there, a bunch of other people. I will list the ones at least that were known to me when we come to this paper. And last but not least, there was the first ever human pig liver transplant. The paper is called Gene-Modified Pig to Human Liver Xenotransplantation by Tao et al. And yeah, this is especially relevant if we talk about the replacement approach of solving aging. So instead of repairing or keeping things young, just like replacing the old parts with new parts. So yeah, first ever pig human liver transplant is relevant to that. Let's start with the first paper. And I hope there are not too many mouse haters watching this. If there are, you can always just skip a study and go to the next one. This one is about the Rodent Aging Intervention Database, short RAID. And yeah, I have a picture of Albie the Gray here because like the mice are kind of his topic. He's really focused on them. And yeah, this database or this study is specifically for people that like to play with data, maybe a bit like me, that are kind of inclined towards data analysis, computers, maybe some AI, ML, whatever, or in general people that do mouse research. First, the paper starts with laying out the problem, and a big problem is that we have a lot of mouse data, but it's very, very poorly organized. And that, excuse my voice, sucks a lot if you want to make new experiments and it's very likely that way that you miss stuff. So you decide to do a new experiment, but it turns out this already has been done. But it's so hard to look through and to like really be sure that you checked all the papers, that this can be like a real problem. Also, it's extremely hard to see big patterns. So if you have a new theory about that something influences biological age and you want to see what the mouse literature says about this, you have to dig through this enormous pile of papers, and you never really know that if you looked at all of them and if there may be something missing that you're overlooking, and it just makes this pattern seeing or like theory evaluating way harder, because you're never really sure that you looked at all the papers. Also, even if you take the time and you dig through the data is still super different, and there are so many different strains, species, sex, age of admission, and all of that you have to take into account, which is very hard if you have to collect the data yourself, and you kind of have to keep it in your head or in your note-taking system or whatever. The solution they propose is the database they built, which, as I called short, RAID, the Rodent Aging Intervention Database. This database contains 121 studies with a total of 212 distinct interventions. The studies all were manually reviewed, and the data was manually extracted from the study. And I put up here the link, so for people watching this, you can just look at the link. If you're listening to this, the link is levf.org/raid. So levf is levf.org/raid. And I'm going to link now. What you can do there is you can select, for example, if you want to have average or maximum lifespan. So let's say we want to look at maximum lifespan, then you want to look at the intervention. You can say, like, for example, let's say I want to look at rapamycin. You can specify an age of onset. So let's say I want to only start at greater or equal than 10 months. An intervention type, of course, rapamycin is a pharmaceutical, so I can just, it won't change anything if I select pharmaceutical, but you can suggest other things like dietary, genetic, surgical, behavioral, cellular, gene therapy, antibody, or multiple interventions. You can specify the sex of the rodents. Let's say I only want males. You can specify the number treated. So let's say I only want like 50 plus. And there we have it. It's only 3 studies left. And you can see in one, there has been a maximum lifespan extension of 188 days, 15%. Another one, 99 days, which is 9%. And another one, 95 days, which is also 9%. And you can also see the amount that they gave. So here 14 ppm in food with a, I practiced this to say this for, but I'm doing it wrong again. It's a carbose. I have to say this later again, and I will do it wrong again probably, but that's okay. Yeah, and the last one with monthly cycles, 42 ppm in food. Just so you can see, this is how you can use their online tool. Let's go back to my presentation. So what data is included? As I already said, species, strain, sex, intervention type. So if it's like genetic, dietary, pharmaceutical, the age of admission, and then the mean or maximum lifespan increase, the control, both of that, mean and maximum lifespan increase for the control and for the intervention group, and also the difference, as I already showed, also including the p-values. How to access the database? They built this web tool that I showed at levf.org/raid, where you can just filter and search for different databases. This is probably the most simple way to access it. Otherwise, they also publish the full data with their paper. And if you want, it's like for if you want to have a study, like if you want to do like some data analysis, you can use their data. They send out a call for, if you find a study or if you know of a bunch of studies that are not included in this database, that you email them. So they add this, and they want to continue updating this database. So it's like a central hub that you can go to before you do a mouse study, or to look at some theory that you have. Look at the numbers, look at the amounts. That's what they build it for, and they want to keep it up to date, that it's not becoming obsolete. Let's go now through the findings that they had when going through this. Of course, it was not the main thing of their paper. The main thing is this database, but they also looked a bit at the data, and the patterns are specifically that the 5 highest median lifespan extensions between 447 and 625 days were all in female mice, none of them in male mice. Also, 9 out of 10 of the highest intervention all include calorie restriction or growth hormone or IGF-1 interference. So yeah, those were 9 out of the 10 highest. Let me take a sip of water, then we continue. Apart from those, VEGF or VEGF overexpression from birth was always in one of the top 3 for all the 4 groups. So males and females, mean and median, and male and female maximum. Yeah, those are the 4 groups, and they're always in the top 3. Also, rapamycin with acarabose. It's very hard if you just read, and I think as soon as I am surrounded more by people that tell me how to say acarabose, I will be way more able to say those hard words. But if you just read them, you never learn how to say them. And the first time you do it on the podcast, it's very hard. But yeah, rapamycin with acarabose was also, I think, in 3 of them was in the top 3. Yes. Last one. In the end of their paper, they made a little note that the data reporting practices of people doing mouse studies was really, or like not really bad, of course. It's amazing that people do this, but they just added some notes of what they would wish scientists to add, and that is, for one, to add early death, so the point at which the first 10% of the mice are dead. Then also they asked for people to not use mean lifespan, but instead of that, median lifespan, because mean lifespan is way too influenced by outliers. And they really asked to please report p-values. So I put the a table from the study up on the slides, and as you can see, the p-value for maximum lifespan was missing in 59% of the studies. So that's like more than half, which is a lot. And they asked to change that and include it in more studies. The next paper comes from the University of California, UC, and it's at the moment only available on bioRxiv, so not published and peer reviewed, not peer reviewed yet. But when I was reading into it, I could see that there's a lot of work in this paper, so I decided to include this here. If you just want peer-reviewed papers, this disclaimer: this paper is not peer-reviewed yet. I think it's probably going to be. You can skip this if you don't want this. I will still summarize it because I still think it's very interesting, and I saw that there's a lot of effort put into this paper. It's actually also the paper that for me took by far the most time to read. It's in total, it's 27 pages. And just for the first sentence, it took me 5 minutes to like really understand. Just to put out, like, the first sentence is not that complicated if you know your stuff, but for me, like, slowly getting into all of this was quite hard. The first sentence was: yeast replicative aging is cell autonomous and thus a good model for mechanistic study for dynamic systems from a dynamic systems perspective. Which I was like, what is exactly replicative aging? Turns out quite easy. Replicative aging is just yeast, they bud, so they don't replicate in as other unicellular organisms do, that they create like 2 equal daughter cells, but they bud. That means they have a mother cell and there's a daughter cell coming out of this, but there's still a clear distinction between daughter and mother. And what replicative aging is the aging of just the mother cell. So when the mother cell is budding, how often it can bud, how long it buds, what happens to it while it buds. That's replicative aging. Second part, replicative aging is cell autonomous. I'm still going through the first sentence. I'll go faster through the rest of the 27 pages, but I just wanted to walk you through what I had to or what I walked through in this first sentence. Yeah, replicative aging. So what happens to the mother cell when it's budding, not to the buds, is cell autonomous. That means it's not influenced by the outside cells. Like relatively, that you can just study the individual cells and you don't have to look at the big interconnection of the cells, and thus a good model for mechanistic study from dynamic systems perspective. So they studied from this dynamic systems perspective, like things influencing each other, and the fact that it's cell autonomous just makes it easier. I have the citation up here if you want to look at the paper. Let's start with the methods. So they choose engineered yeast. Why did they choose yeast? Because they have a short lifespan, so it's relatively easy to study. They are genetically tractable, so short genome. They have an okay resemblance with multicellular organisms, so they also show many of the hallmarks of aging, which makes them a good object to study. And they, the fact that replicative aging of yeast is cell autonomous, so it's more simple than if you have to study an interplay of many cells and how they influence all each other. And this way you can just look at one cell and there's enough stuff that is influencing each other in one cell that you have to think about. I said that those yeast are engineered. How are they engineered? They're engineered with a daughter arresting program, which means that the daughters that they bud, they are not able, like they kind of go into stasis right when they bud. How does it work? It works by genetically engineering that one of their promoters is repressed by the presence of glucose. So if you put them on glucose, one of the promoters is a region in the DNA that kind of starts transcription, so it can later be translated into a protein. And if you repress this promoter, this area doesn't get, the following area doesn't get transcribed, doesn't get translated, and you don't have the protein. And this promoter is in front of a region that codes for an essential long-lived membrane protein. And what they do essentially is they grow those cells in a glucose-free environment. So they're just normally expressing this, promoter is just normally working. They are just normally expressing this long-lived membrane protein. But then during the experiment, they put them on this glucose media. So all their daughters, which because it's budding and not like this equal cell separation, the daughters don't inherit the membrane of the mothers. The daughters won't have this membrane, this like critical membrane protein, and then they just can't grow, can't replicate. In that way, it's easy to just study the mothers, and you don't always have to do like this kind of cleanup or whatever with bringing the mothers. You can just like let them bud, and you don't have to worry that the daughters also start budding and you will have like this explosion of super many cells. That's the daughter arresting program. Then what they did is they used a high throughput microfluidics device, which has, like I put on the slideshow, there is a picture of what they had in their like a figure of this device. It has 32 chambers, which each of those 32 chambers is like 32 times like those triple chambers that you can see, but it's just one that is relevant. Has 220 individual micro cell chambers. It's what you can see in like the lower here, the lower one. So that means you have 32 times 220 cell chambers, and in each cell chamber there can be one yeast that is trapped, and you can have like each one you could theoretically put a different strain in, and make so 32 different experiments per microfluidics device. So they made this with thousands of mice. Did I say mice? I'm still two experiments, two papers before this. I mean, of course, yeast. So they did like thousands of yeast at once, and that way it's like really high throughput. They had a camera on top that took photos every 20 minutes, like a microscope, and it took 17 Z-stack images. That's also a thing that I was like, oh, what's that? What's a Z-stack image? And maybe super clear to you. I will still explain it if it's not. 17 Z-stack images means that there were 17 images taken from with different levels of focus. So the focus like shifting from down or up to down, so that if you have like a three-dimensional object, sometimes like in the beginning maybe the top of the three-dimensional object is in focus, then a bit lower. Like I have like a camera, you can see like now the focus is very clear on my finger, and now the focus is on my face again. And you have to imagine that there are just 17 layers. There's like one where's my nose in focus, one there's my eye, one there's my ear, one there's the wall behind me, one there's something in front of me. So that's like a standard practice to just use those Z-stack images. Let's go to the methods. As I said, they used the yeast. They used the high-throughput. We were already to methods. Sorry, high-throughput microfluidic device, and then they took the pictures and they tracked. What they tracked was the organelle and cell size and shape. They tracked the cell cycle time. They tracked the activity of different cellular processes, so proteostasis, genome stability, mitochondrial function. That's what they tracked. How did they track it? They track it through fluorescent reporters. And they said the yeast were engineered with the stored arresting program, but not the only thing that they engineered about them. What they also did is that they changed some of their genes, some of the proteins they express, to also express on the end of them a sequence which codes for this fluorescent reporter. So on the C terminus, so one end of the amino acids chain, which is a protein, they like put this fluorescent reporter, which then signals how much of this protein is there. So if you take this picture and there's a bunch of this fluorescent reporter inside the cell nucleus, you know, like, okay, this thing that I marked in this specific yeast strain is overexpressed right now. And same with like many different proteins. They did it for many different things. How did they make sure that this was done on all the yeast successfully, and they didn't have just some yeast where this genetic engineering didn't work? That's something that's really cool, I think. And it's that when they put this new gene in with the fluorescent reporter piece, they also additionally added a kanamycin resistance gene. Kanamycin is just a thing that kills bacteria, or like kills yeast. Sorry, not bacteria, yeast. And after they added this, they put the yeast on a kanamycin plate, where essentially all the yeast that didn't get this kanamycin resistance gene died. And the ones that got the kanamycin resistant gene, and with that also the additional C terminus fluorescent reporter, they didn't die. And that's such a cool way. Like, I'm fascinated by what biologists can think of to figure out how to make sure that this gene editing happens, works, that the fluorescent reporter is actually there in all the cells and not just in some. They just add this resistance gene and let the other ones die because they don't have the resistance gene. In total, they used 34 single reporter strains, so strains where they only edited one gene, that they have one protein that also expresses this reporter on the C terminus, and 29 double reporter strains, so where they had two genes, so you can see like the interaction. For example, you could have one that marks exactly where a specific cell organelle is, because this protein is just expressed in one organelle, and another one that you're interested in how much it is in this specific organelle. And if you don't have both of them, you don't know exactly where example where the specific organelle is, and if the other thing is expressed in there. And yeah, that's why they also used double reporter strains. What they then did is image segmentation, and I was like, yeah, that's the part for me. I know stuff about image segmentation. That's going to be easy, but it was definitely not easy. It took me quite a while to understand this. Essentially what they did is they used a pre-trained UNet to figure out the cell boundaries, and what they also tracked was the distribution of mitochondria. So where exactly, like what shape the mitochondria had. So they could be like, for example, round, that they're like outside. They could be like completely like on the outer side of the cell. They could be like distributed. They could be like in patches and whatever. And to classify this, they used the pre-trained ImageNet model. So ImageNet is like a giant data set that all the machine learning people were like trying to figure out how to make image classification. So like, oh, this is a dog, this is a chair, this is a cat, whatever. They used a model that was trained on the ImageNet data set, a pre-trained model, and then specified it to classify the different mitochondria distributions. They also used a bunch of other image segmentation technique that I decided I don't want to bore you with, because if you're really interested, just go to the paper and read them. There's no point in me listing all of them, but yeah, just those two for you. Then for data analysis, they used Granger causality. So that means that if you have two different, wait, let me drink something, my mouth is dry. Granger causality, they essentially aligned the levels of two different proteins. As I said, like with the fluorescent promoters, you can easily track how much of each protein is in there. And then they looked through Granger causality if the level of one predicts the level of another. That's one thing they used. Also, they correlated marker intensity at different points of life with the cell lifespan. So is there a specific point in time where the amount of a specific marker predicts the lifespan of the cells? Now let's go to the results. First of all, the first result, and the most obvious one, is that the cell size predicts the cell age. So essentially, the bigger a cell is, the older the cell is. Cells get bigger with age, and this is a linear increase. Second, this also happened to the nucleus. The nucleus also increases with age, but there's a difference. In the beginning, to like mid-age or something, the nucleus increases proportionally to the cell size, which is also linear, and at some point it kind of breaks out, and then it gets non-linear. It gets faster, and this, I don't want to spoil you, but yeah, this is also something which is related to the cell. I don't know, we already at results, so I can tell you, which is like for cells that this happens earlier, so it kind of breaks out earlier, the nucleus starts growing faster than the cell size earlier. Those cells also tend to die earlier, be shorter lived than the cells that keep this linear growth of the nucleus for longer. Yeah, because in general, like the nucleus size is a big thing for how fast the cell dies. You can also see this very clearly in the figures I put down here, that like for nearly every cell, it starts out with like a relatively small nucleus, and before death, the nucleus became relatively big, and it, like even way more clear it is with the cell size. Third finding, the nuclear proteasome, so that is the first of all, the nuclear proteasome concentration at mid-age predicts cell lifespan. What is the nuclear proteasome concentration? So nuclear in the nucleus, proteasome, that is the cell machinery that kind of garbage collection picks up broken, ubiquitinated proteins, so proteins that are marked for trash collection. And yeah, the concentration of this at mid-age predicts lifespan. So high is good, low is bad. And when they were looking into this, they figured out that the because this is just for nuclear protein concentration and not outside. And they figure out that the nuclear size mediates this, because the idea is essentially that when the nucleus gets bigger, this is a kind of a ball. And with the ball getting bigger, the surface area of the ball increases slower than the volume of the ball. So if all the proteasome components have to enter the nucleus through the membrane, and the nucleus is growing, at some point you have a way bigger, like way smaller surface to volume ratio, because volume grows to the r cubed and surface only r squared. Like, of course, yeah, formula is different than just r squared, but essentially it's like one grows squared, the other grows cubed, so to the power of 3, which lets it grow just faster, and so the ratio is off, and at some point there's not enough which can enter, and that causes this dilution, which causes the nuclear proteasome to be low. The fourth finding they had is that when the mitochondrial distribution was circular, so like more on the outside, that this predicts a longer lifespan. Then they ask question, does proteasome expression affect lifespan? So it's what I already said, that like proteasome expression, what they found is that it definitely does increase, like it does predict lifespan, but is it causal? So what they did is they overexpressed RPN4, which is a promoter that, I already explained what promoters are. It essentially activates transcription of all the all the proteasome proteins, and they build it in a way because RPN4 is actually, it's a transcription, I think it's it's a promoter, like a protein that lets this promoter work. A transcription factor, that was the word I was searching for. And the problem with this transcription factor is that it has like a degradation signal, which makes like a feedback loop that overexpresses very hard, because the more you express it just gets degraded very fast because it has this degradation signal. But what they then did is that they removed this degradation signal and created so RPN5 is this transcription factor, which helps to start the transcription of the proteasome components. But RPN5 has on itself this degradation signal, which they removed in the RPN5 that they put into the cell. And then they put this RPN4. I said 4, but I mean 5. I said 5, but I mean 4. Sorry, it's getting late. Last one I record for the day, the rest I do tomorrow. They put this in front of an estradiol inducible promoter. So essentially, if they put estradiol into this microfluidics device, it activates the promoter, which then lets it transcribe the RPN4 without the degradation signal. Then this gets translated. Then it activates the transcription of the proteasome components, and you have more proteasome at the point where you induce estradiol. Yeah, very cool. That's all of that. That's why it took me so long to read this paper. Turns out, when you do this, actually the nucleus grows. And as I already said, nucleus growth is like higher with older age. So like that's curious. Another thing, mitochondria are distributed more circular, which is good because, as we heard, circular mitochondria are kind of correlated. We don't know if it's causal, but it's at least correlated with a longer lifespan. Third one, no lifespan change. So yeah, it didn't increase lifespan, even if you have more proteasome. And the question there is, maybe cancels out. So because it increases the size of the nucleus, is it possible that this just the fact that it increases the size and at the same time it also increases the amount of proteasome, like those benefit and the non-benefit cancel each other out. This is maybe a possible explanation. But also another possible explanation is just like, the proteasome is not the only factor that gets diluted with growing nucleus, what I explained with like the size. There are also a lot of other factors that get diluted through this size difference of the nucleus. And then it's clear that, it's possible that just overexpressing proteasome doesn't do as much good as one might want. Let's go to the discussion. What they set out to be like from their experiment, like the aging drivers that they found to be relevant was one, the loss of proteostasis. As I said, the proteasome overexpression, which they tried, the proteasome, which is like clearly the main thing that they found to be correlated with cell lifespan. And the second one is that the dilution of nuclear proteins probably has an effect on cell lifespan, which would also explain why, as I already said, protein overexpression doesn't increase lifespan, because only increase overexpression proteasome maybe fixes the proteasome problem, but there are a lot of other nuclear proteins that get diluted by this growth of the cell nucleus, where at some point the membrane is way bigger, or the main membrane to volume ratio is just off because r cubed grows faster than r squared. That was paper number 2. I'm gonna do a break. This is way more exhausting than I thought. I was like, I'm gonna easily hit those 5 papers, no problem. But turns out that between the last one and this one, I was just like laying on my bed and was like, I'm doing great. I'm putting the effort. I hope I'm presenting this well. If you want to give feedback, I like that. But it's exhausting to be like sharp and to explain it clear on the point as I understood it. So I'm gonna do the rest tomorrow. For you, it will be just in a couple of seconds, but for me, see you tomorrow.
New day, new paper. This one is about optimizing metabolic health with digital twins. And before we start, I want to show you something, because actually it's now in the morning and I was planning to be in the library, but the library was closed. So right now, Mark, a friend of mine, is sitting behind me and he's doing his work, but he has white noise on his headphones, so he won't be bothered by me recording this. So let's start. Optimizing metabolic health with digital twins. You can see the citation here for people watching it. What they say in this paper is that they propose a possible solution to the increase in cardiometabolic disease that we have currently. What is cardiometabolic disease, CMD? That includes metabolic syndrome, so obesity, all that jazz, type 2 diabetes, and cardiovascular disease. And this cardiometabolic disease has shown a crazy increase in the last years, and they propose this digital twin solution to be like a personalized healthcare method to solve this crazy increase in cardiometabolic disease. And I chose this paper because I think this idea of digital twins, even if it's still super rudimentary, and maybe even if this specific metabolic health digital twin, metabolic flexibility digital twin that they propose here is not the be-all end-all amazing thing, I think the idea of metabolic twins is just not metabolic twins. Digital twins is a very good idea, and we should definitely pursue this to personalize healthcare further and further, and build amazing digital twins. And that's why I chose this. And let's just jump right into this. So, metabolic flexibility. That is kind of what this paper spins around. I said they like the paper is called Optimizing Metabolic Health with Digital Twins. How do they quantify metabolic health? They quantify it as metabolic flexibility. So metabolic flexibility is the ability to switch between different fuel sources. So different fuel sources, glucose and fat, those are the main two fuel sources that we use. Like there's also protein, but like the main two things that we use is glucose and fat. And so usually you burn, after you eat, let's say you eat some pasta, you definitely burn glucose for the next couple of hours. And then as you don't eat any carbs, or as you don't eat anything, let's say you go to sleep, you slowly, gradually stop, like go down with the glucose metabolism and go up with the fat metabolism, which is also then called ketosis, because the fat gets broken down into those ketone bodies that your body uses. And this switching is an indicator for metabolic health. So metabolic flexibility is how fast you can switch between fat and glucose metabolism. What is interesting about it is like very strongly correlated with fitness. So athletes have higher metabolic flexibility. It's also correlated, like negatively correlated with weight gain. So if you have bad, low metabolic flexibility, it's way harder to lose weight and more likely for you to gain weight. But at the same time, it is not a perfect correlation with body weight. So it is possible for a person to be overweight, but at the same time have a good metabolic, like a high metabolic flexibility, which would tell you something more than just the person is overweight. Because if you say, okay, all overweight person are equally unhealthy, that is not true, because a person that is overweight but has a high metabolic flexibility might be even healthier than a person that is not overweight but has a low metabolic flexibility. So it's not as clear-cut. Of course, it's more likely for overweight people to have worse metabolic flexibility, just because it also makes it more likely to gain weight if you have a low metabolic flexibility, but it's not a must. And I think that's just important to mention. How did they propose to measure or how is metabolic flexibility in generally measured? It is measured by the time, or how they propose in the papers, the time of days that it takes for you to switch to fat metabolism, so ketosis, when you follow a specific exercise and diet regime. So they tell you exactly, okay, this is what you can eat, this is what you do, and then how long in days it takes you to switch from burning glucose, which is kind of the default state with, if you follow like a relatively standard, not ketogenic diet, and the time it takes for you to switch into ketosis, if you kind of follow a ketogenic diet, maybe even fast. Like they don't specify the exercise or diet regime, but this time of days that it takes for you to switch is your metabolic flexibility. And like, let's say if you switch in like 3 days, it's like very good. If you switch in 10 days, this is like rather poor. Or if you don't even switch after 14 days, this is like extremely poor metabolic flexibility. But how do you measure your fat metabolism or your glucose metabolism? Like how do you know that you have switched to ketosis? And for that, they list a couple of solutions. So the first one, that is the respiratory quotient. So how that actually works is that you kind of have a device that you blow into and it measures, or that you breathe into, and it measures the ratio of CO2 that you produce to the amount of O2, so oxygen, that you consume. And depending on if you're in glucose metabolism or fat metabolism, you burn, like this ratio is different. So if you burn glucose, I think it's like around 1, and if you are more in ketosis, this is at 0.7. So from this like analysis of your breath, you can see which of the two fuel sources you are mainly using. And yeah, but the thing what they say about this is that the problem with this measurement is like you need to have this device, you have to go like where the device is, so it's like kind of not good to do in large scale. So what they propose is that you measure which metabolism you're using through blood measurements. So that's also the gold standard for a ketone body in blood measurement, is that you kind of do those finger prick measurements with a device that measures the amount of ketones that you have in your blood. And for glucose, of course, they are like those sensors, those continuous glucose monitors. I actually tried one once that you can put into your arm and that measured the amount of glucose in your blood. But the main important thing is the ketone measurements. They also propose a bunch of other measurements that are possible for ketones, but that are, or for if you're in ketosis, but they're not perfectly validated right now. And like this, what I said with the finger pricks is the gold standard, which really say, okay, yeah, this is what we would use. But if the other things get better validated, that would be of course amazing, because it is not as invasive that you have to always like prick your finger and get some blood out to measure your blood ketones. What the other things that they proposed, that was once from your sweat, you can measure from your urine, you can measure from your breath. There are epidermal sensors, so like similar to the continuous glucose monitors. And also you could maybe estimate from CGM data, so continuous glucose monitor, what I was talking about, what diabetics and sometimes people that want to optimize their health have. From that, you might also be able to, or they have people that did this, that they estimated their blood ketones from the CGM data. But this is all, like all the measurements that I just said are still like not perfectly validated. And that's where they say, okay, this finger pricks is the best way to measure blood ketones at the moment, and that's also what they propose to measure if you already switched. So what you would do is you do those regular measurements to see how many days it takes. And from that you calculate, or the digital twin, the AI calculates how high your metabolic flexibility score is. So now let's go to this idea of the digital twin. What is this idea of the digital twin? So in general, a digital twin is a digital entity that changes in a way that reflects a target object or process. In the case of this specific digital twin, the target object or process, that is you, or the person that is the user. So essentially it's an entity, a computer program that tries to model your metabolic flexibility in response to different behaviors, in response, like, okay, well, how would your body react to different things, and how is the long-term outlook? And the idea of digital twins is already used a lot in diabetes management. So what happens if you eat this specific thing? What happens if you do this specific thing to the blood glucose of the diabetes patient? Now specifically, metabolic digital twins, what they propose is, this model would take in your glucose and ketone measurements. So as I said, like finger pricks or continuous glucose monitor, finger pricks for ketones, continuous glucose monitor for glucose. It takes in your diet, it takes in your exercise regime, and it takes in what they were not specific about, a bunch of other health measurements. So you could think of blood tests, you could think of blood pressure, you could think of maybe respiratory quotient, and respiratory rate, heart rate, like all of that. You could feed it in there, but that's not the main thing. Like the main thing that they focus on in this paper is glucose, ketones, diet, and exercise regime. And what it would give you is that it calculates your metabolic flexibility score. So let's say between 0% and 100%, like how good your metabolic flexibility is. It would output diet and exercise recommendations, and it would output future health predictions. So like if you keep following this way, this is what is kind of gonna happen to you. Yeah. I wanted to add some personal notes that I think, like when I was reading this paper, because as I already said, I love this idea of digital twins, but the idea to use metabolic flexibility as the main measurement that you're optimizing for, maybe it's a very good measurement, but at the same time, a measurement that takes multiple days. So let's say I take 7 days to switch. Then I have to follow the exact regime that is made for the testing for 7 days, and to just have one measurement. And I think you can already see that this doesn't seem so scalable. So if I have to do that, like even me who really likes to work on his health and like kind of optimize those small things, it's like I would have to, if it takes 7 days just to measure once, like I see that I wouldn't measure that so often. So like maybe once a month max, and that would already be, I think, way too much, because I want to follow the health, exercise and diet regime that is optimal for my health, and not that is optimal for measuring. So if I have to measure only once a month, which is very little data points, if I have to, okay, actually maybe it's okay to actually do it also every half year, is to see like if the general trend is right, but kind of more data is better. So if it's so hard to get one measurement, and for this measurement I even have to change my whole diet and my exercise regime for 7 days, I think the cost is way too high, and I would love to have another type of measurement. So maybe an approximation of metabolic flexibility from a continuous glucose monitor, maybe still regular ketone checks, the invasive ones, like the finger pricks, but even like maybe wait for continuous ketone monitor and then estimate your metabolic flexibility from how day-to-day actions impact your ketone and glucose response. And I guess it should probably be possible to use some AI to estimate this. So if you see your body's responses to eating, like let's say an apple or something, you can probably estimate a lot from this. And even if you can't use metabolic flexibility, I think we probably have better measurements. And even if metabolic flexibility is a very good measurement, just the fact that it takes so long to measure, I think makes it not as viable as a perfect measurement to use for this. So the next paper is about how cities, communities and clinics can be perfect test beds for human exposome and aging research. This paper was co-authored by a bunch of known people or of the longevity industry. So to just name some, it's Neo Baseline, Andrea Meyer, Alexei Strigen. Alexei and Andrea were both on my podcast, actually, previous episodes. Nikhil Yadala, Evelyn Bischoff, Brian Kennedy, just to name a few. And the key idea of this paper is that we should study the exposome, and environmental factors that influence health, and actually how they influence health, and that cities are just the perfect place, like cities, communities and clinics are the perfect place to do those large-scale exposome experiments or studies that is just not utilized right now. Like, there's so much sleeping data kind of that we could use by tracking and intervening to improve the health of a bunch of people and learn so much more about how our body health, our health works, and what are the different influences that are important that we're maybe not aware of right now, and especially not aware of how much what influences what. So the premise of this paper is that our current model of healthcare is kind of unsustainable, because what we do right now is we wait till somebody gets sick and then we react and then we try to do something about it. But this just creates way more people that need healthcare at a situation where it's way harder to intervene. And they suggest that we need more prevention and less reaction. So do more things before to help people to not even get sick, and that way improve health instead of waiting for somebody to get sick and then do something about it, because this is, as they say, not sustainable the way we do it. And what they then suggest is what they call the Human Exposome Project. And I like the wording. It's kind of a bit like the Human Genome Project, where people got together and were like, we have to sequence the human genome. And now is the Human Exposome Project. So we want to get together to learn about what the exposome does to humans. So gather data to figure out, gather as much data as possible to figure out how the exposome influences the human body. It's like an initiative to understand, the exposome influences on health and aging, especially also on aging. But what actually is exactly the exposome? So the exposome is the set of environmental triggers that, a set of environmental triggers that can cause, for example, age-related changes. So environmental triggers can be everything like your air, the quality of the air you breathe. It can be how much greenery you have around you. It can be like what you eat. It can be actually I also put this slide up. I could just look here. So you have the external exposome. So specific things like your diet, the pollution, your environment, the chemicals you're exposed to, your lifestyle, the amount of light you're exposed to, what times you're exposed to the light, what type of light. Then the general things which are population-wide, which are relatively similar within a city or something, is the climate, the economic status, that's probably in the region of a city, the education level, the culture, those things. Then this is kind of what feeds into your body and then changes things inside your system. So there are molecular and genomic interactions that can activate pathological pathways in your body, and then they change something. So you're born with your the genome and everything. They call this actually internal exposome, where it's in that case you would say everything is kind of exposome. So what you are born with, even your genomics, transcriptomics, cell count, proteomics, metabolomics, methylome. This is more measurement, but as you can think of it as actually what they call or what they present, which was before already done, the blueprint theory that environmental triggers cause age-related changes. So let's say I breathe polluted air, that would trigger molecular and genomic interactions, cascades that activate those pathological pathways, and therefore then reduce my overall health and just trigger those age-related changes, which wouldn't have been triggered if I wouldn't have those pathological or this environmental triggers polluted air, for example. Of course, you can never be zero environmental problems, even unpolluted air, it will trigger pathological pathways. But that's exactly what we want to figure out, what are the things that trigger age-related changes, and then figure out how we can change them to at some point have it just decrease the amount of bad environmental triggers that we put into our bodies, and that's what this Exposome project is about. Of course, they don't say that this is the only thing that causes aging, which is definitely not, but it is definitely a thing that plays into, and a knob that we can tweak quite easily, what you put into your body, the amount of pollutants, chemicals. This is something, it's quite a low-hanging fruit that could improve health and longevity for a large number of people. They compare the two approaches to improving people's health, which is the classical and exposome approach, or studying how to tackle aging and disease. The classical approach includes we study small molecules in the body. What do they do? So we look at this specific small molecule. If you eat it, this does this to this other molecule, and this happens, and then the overall effect is that something goes wrong, for example. And this would be the classic approach, but the exposome approach that they advocate for in this paper is that you study how specific environmental impacts, your specific environment impacts health outcomes. So if you see you're in an environment where there's a lot of greenery, I will come back to this a couple of times, that it might increase your overall health and might just create this positive feedback. Now, what they propose is that cities are they could perfectly track the exposome of citizens, or at least like partially. So with advanced technology like measuring air quality, measuring water quality, measuring a lot of different things, pollutants, amount of traffic, greenery, citizens, like crime rate, so like for maybe stress, overall stress in the population. That's like all the things that could be easily tracked in a city, and then they could also track health outcomes, so like hospitalization rates, all of that, to learn about what influences the health. And there's just so much sleeping data there that we could use to understand how different factors in a city influence the health. And we could just build this huge knowledge bases. Like there's just so much data that we're not using right now. Another thing they propose, which I found interesting, is that cities could track the amount of drug residues in water. So if you take specific drugs, you could, this shows up in the urine, and if a lot of people take drugs, this shows up in the wastewater of the city. And if cities track this, they could react early to like some increases of drug use. They could send city councillors, they could help like the population in that way to see, okay, this area, like there's an increase in the use of a specific type of drug, and just react faster to help citizens' health. Also, cities could run exposome intervention experiments. So I come back to the greenery. It's just, for example, in one of the streets of the city I live in, they just put up those huge buckets with trees in them, like giant buckets. And now suddenly, there's like within a day, the city had like trees all along. Before it hadn't. And stuff like this, like you can make a city green with a snap of a finger if you take the money if you have the money and want to do that. And you could see how this influences the health of people living in that area, for example, how this increases like different factors that you care about, like clinical outcomes, overall like the health of the population of the city. And there are already some cities that are pioneering this, like longevity cities like Rotterdam, Barcelona, London, where people dedicated to try to improve citizens' health, like really watch the exposome of their citizens. The problem now is that it's hard. You can't publish the hospitalization data of your city because this is private data, and it is not just like, all of that is nothing you can just freely share. But around that, they showed like a really cool solution that I really like, and I think we should make way more use of, is that the publishing of synthetic data. So if you have this data set of your citizens of a city that you are not allowed to share because this is private data, what you do is you just train a model on this to create synthetic data. This is used a lot. Like this is just the standard approach for data augmentation. So if you have too little data, you can use a data set and create more data that comes from the same distribution from this. But now what they propose to do is that you create this synthetic data from the distribution that has the same distribution as the core data, and then you just publish this synthetic data, which means the synthetic data is not like there's no individual point that was before in the synthetic data, but it follows the same distribution. So it can be like the same type of analysis can be done on this, and this data can be freely shared, which completely like reduces this privacy issue, and it would just enable this broad sharing of data with researchers, which could you don't expose personal information, and researchers can use this to learn, analyze the exposome, like what we learned about the exposome. how could we use this exposome? They list a bunch of things that it could be useful for. It can be used to create better biomarkers. It can be used to improve intervention and recommendations. It can be used to improve policies. It can help companies, investors to make smarter health decisions. So if you see like, oh, this thing has a strong impact on health, just there are so many things that can be used if we properly collect the data of this broad number of people, including the exposome. What they use as a model, or that I also find really interesting, is health IDs, which is already used in India. That is, like, you have this ID, which is a common place where all your health data is stored. And of course you have to be careful with this because this is a lot of centralization. But if you implement this in the right way, then this can be super useful, because that way your health data is not scattered everywhere, but there's a central place where your health data is stored. And with the permission of the citizens, this could be an amazing place to use to learn about how different things influence health. Especially if you also add exposome data. So, for example, the city you live in, the air quality at the time you live in there, and you can see, oh, okay, you could do such large-scale studies if you have this giant amount of health IDs of people, and you know, okay, people living in that city are just less healthy than people living in that city. Why is that? Okay, this city has more greenery, I don't know, or different factors. Of course, it's going to be super hard to learn, but just having this huge amount of data could improve so much. Like, data is such a big bottleneck right now, and there's just so much sleeping data that we could use and that we could learn about, especially also if cities do experiments and it's not only correlation, but we actually learn about causal effects. So, last paper, and this one is going to be a quick one. It's about the first ever pig human liver transplant. The paper was published by a Chinese team. It was published in Nature, and it's called Gene Modified Pig to Human Liver Xenotransplantation. The recipient was a brain-dead patient, and he got the liver of a 6 gene edited pig liver. So, and the liver functioned throughout the whole 10 days. 6 gene edited pig liver. This is because the problem is, if you transplant from, like, there's already this problem if you transplant from one human to the other. If you transplant an organ from one human to the other, the problem is that there is very likely this immune reaction, because this is not your liver. Like, your body, like, whenever you get an organ, you most likely have to be placed on immunosuppressant drugs that fight off this, to prevent your immune system from attacking this foreign body that is now in your body, this foreign organ. And this problem is already there if you transplant from humans. But if you transplant from another animal, this is like way bigger of a problem. So that's why they edited some of the genes of this, like 6 genes of this pig. So the main things that the immune system reacts to of humans get like taken out. And this actually worked. Of course, the patient still had to be put on immunosuppressant drugs, but still this is like a big success that this worked for the first time. And the amazing part of doing it from a pig is that we have this immense shortage of organs right now. And there are people thinking about, okay, how can we grow new organs in like the lab? But this is so hard. And nature kind of already figured this out. So pigs have very similar organs to us. And if we manage to build pigs that have like perfectly good organs, the replacement idea of longevity, like, okay, instead of repairing everything, instead of figuring out how it works, no, I just take out some of my organs and put in new ones, like the ones that are breaking down. I take out my liver, I put in a new liver if my liver is not working anymore as I want it to work. This idea comes way closer, because with humans you have to wait till somebody is like in the right type of accident, in the right type of moment. This is also an organ donor. Like, this is absolutely not scalable. But with pigs, they would be extremely scalable. Like, if we really figure this out, this would mean like no organ shortage at all anymore. So the pig human liver transplant, what they did was a heterotopic auxiliary transplantation. And before, when I read this, I was also like, a heterotropic auxiliary what? Like, I don't know what those words mean, but of course now I know. And so what exactly this type of transplantation is the heterotopic means that it was put on a different place. So instead of taking the old liver out and putting the new liver in, this liver was put into a different place. Auxiliary means supporting. So it kind of supports the failing organ by putting it somewhere else. And yeah, this is like actually a very, very common technique. The problem is, like, at some point you probably like this way you could actually, like, if you manage to fix the original liver, you can just take the new one out again. But this it's just like, this is the type they used, and it's especially useful if you are in transition periods. So if you await a full transplant, so if you're actually waiting for a human to have an accident and you get his liver, then this can help patients to survive the waiting time without having to sacrifice their own liver. And it's just like an intermediary step, but it's still like amazing that we were able to put the human, like the pig liver into a human, even if it was not put at exactly the place where the human liver was. So where we're at right now with pig to human transplants. What has already been done also before this paper is that a heart got transplanted, a kidney got transplanted, and now the new thing is that a liver got transplanted. And this is amazing. Like, we already got like 3 important organs, and just the better we get with this, this can be immensely valuable and solve this incredible shortage of organs that we have right now. On the screen right now I have the full citation of this last paper, so if you want to read in more detail how they did it, it's very technical and it was very hard for me to read. You can just look at the paper. It was written, you can just look at it, click at the link, copy the link or something. So that was the first edition of the longevity news. I had a lot of fun doing that. It was also very hard doing that. I don't know, if I reflect now on it. I want to learn as much as possible about the longevity industry, and I think what I did now is like I increased my horizon, like my view of the field by a lot. But at the same time, it's super hard, especially in the beginning. If like every paper that you read is like a completely new world for you, it takes forever to read a paper. Like, I think especially on the second paper, it took me like 4 days to like really understand it. And it's like, this is a lot, and I also have a lot of other things that I want to do, and those are 4 days that I'm not coding. But at the same time, it's like, it's such a, I learned a lot from this, and I will definitely do the one for May. I already prepared some things for this, and I will see if I continue. Maybe if you can write in the comments how you like it. I would love to have some feedback. Thank you very much for listening to this. I hope I could provide some value to you, and see you in the next one.